All right. Thank you. Thank you all for joining. Welcome, everyone. We have to wait on. We have waited a few minutes on all participants, but possibly a few more will join later. But we thought to start about 5 minutes over past two. So my name is Eddie van Bremen. I'm a sales representative for Pemgene for a few years now. And we thought, hey, let's set up a few sessions to get back in touch with our customers and share our best practices that we have learned over the years. And we want to also update all of our customers in multiple sessions. And this is the first of hopefully many. In this first session, we also kind of introduce our new colleague, Peter Linders, and he will try to educate or update all of you with some of our best practices. We chosen that everyone is muted and don't have a webcam on to make it less messy. But if you have a question or a remark, please ask us. But you could, for example, type it in the chat, and then we will try to respond to it at the end of the whole presentation. So hopefully we can address all of the questions, and if we can't, we of course get back to you in a next session. So Peter, yeah, maybe you can quickly introduce yourself, and I'm looking much forward to your presentation.
All right, thanks Eddie for the introduction. So welcome everybody. I am Peter. So I started back in end of July at Pemgene, so I'm relatively fresh. But here I am today presenting some of our best practices to you guys. So I finished my PhD and then I moved into becoming an application specialist for Pemgene. So I've studied how Pemgene does things recently, and I'm sharing this information with you. So to give you an overview of what I'm showing you this session, there you can see that on this slide. So we'll start by talking about how to find new users for the platform, as well as some more specific assay-related info. So you can think about optimizations for your experiments or how to design your experiments properly. Typical results you can expect by using the platform, mostly on peptide level. So more detailed analysis will be a topic of a future session. And I also include a section on frequently asked questions, especially for finding new users or experimental design. That will be towards the end of the presentation. And finally, I'll give you a little sneak preview of the next session, which will be about reporting of the data. But before we get into the whole meat of this presentation, I would like to share the animation that some of you might have seen. So let's take a look.
We are Pemgene, and we specialize in measuring kinase activity. In this clip, we explain how our technology helps to understand cellular signaling. Let's start at the beginning. Cells are the fundament of all living things. They are the drivers of growth, adaptation, and defense. Cells are continuously responding to external stimuli. Upon stimulation, a signal is passed through the cell membrane to the nucleus, and this leads to genomic and phenotypic changes. Kinases are key players in transducing these signals. Studying kinases is therefore of great importance to fully understand changes in cellular processes. Kinases are enzymes. They transfer a phosphate molecule from ATP to a specific phosphosite on a target protein. This is known as phosphorylation. Phosphorylation leads to changes in conformation and can make kinases more or less active. Activated kinases will start to phosphorylate their targets, leading to a cascade of signals, also known as a signaling pathway. There are two major types of protein kinases. One targets tyrosine, and the other serine threonine phosphosites. We developed an array with peptides mimicking the in vivo phosphosites. This allows to measure activity changes of kinases or pathways in the lysate. When a lysate is incubated, the active kinases in the sample start phosphorylating their targets on the array. The amount of phosphorylation is visualized with a generic fluorescently labeled antibody that binds to all phosphorylated targets. A common application is comparing treated and untreated cells to look at the differences in kinase activity. We interpret which kinases are responsible for phosphorylating the targets that are significantly different between the conditions. The functional and multiplex properties of our chip allow us to display cellular signaling. And connecting these kinase fingerprints to a clinical response could lead to the identification of a predictive biomarker. Some other interesting features. Our technology utilizes the full-length kinases that are present in the cell lysate. A single fluorescently labeled antibody is used to visualize all phosphorylated targets. Our technology is sensitive, capable of measuring from 0.5 microgram of protein input. The majority of the kinome is covered by our multiplex assays. As fellow scientists, we are curious how our platform can make the difference in your research.
So the scientist in the animation was actually modeled on me, fun fact. But to continue the presentation, now we are all on the same page. I would like to refer to the resource pages. So I'm guessing you're all familiar with the PAM Station 12 resource page if you've used the PAM Station system recently. But we now also have a PAM Station 12 core facility page. So the slides that I'm showing you today will also be on that page, as well as a recording of this presentation soon. As well as some more core facility specific information will also be at this page. And we'll make sure to share this link with you as well, so you don't have to write it down necessarily right now. For the rest of the presentation, I would like to stress that much of the information will probably be familiar to you at in some form. But I hope there will be some new information for you as well, or some new understandings that you hadn't had before. So please stick around until the end. So as Eddie mentioned, I'm just recapping. Questions can go into the chat, and then between each section we can discuss about these questions. So moving on to the first section, this is on how to find new users. So of course, if you have the PS12 system, you want people to run on the system because otherwise there is no use having the system in the first place. And we've noticed that in some cases, it can be difficult to find new users. So here I would like to show you some approaches that we take to find new users, both from the academic side as well as the industry side, which can also can be very interesting for you. So roughly we can split our users into three categories. So on one end we have fundamental groups. So these are groups that are mainly performing fundamental mechanism of action studies. So they look at understanding how certain pathways are activated or inhibited in different models, or they try to discover entirely new pathways. Next to this, they also attempt to understand pathophysiology using different disease models. So these are generally model systems. You can think about model cell lines or model animal models. And these are typically quite small experiments. So these are maybe one-run experiments where you use 3 chips and just a couple of conditions. So the sample size is pretty small. And on the other hand, we have clinical groups that do experiments using human material. And in this case, you can consider, for instance, the discovery of certain biomarkers to stratify patients, or to discriminate responders versus non-responders to a certain drug. And we also, if you might be familiar, if you're familiar with our diagnostics platform, this is also an avenue that we are approaching to really use like kinome profiling as a way of diagnosing clinical conditions. So compared to the fundamental research group, clinical research groups have many more samples. So these are generally upwards of 50 to 100 samples. This also has to do with the large variability that there's found in human material. And of course, special considerations need to be taken in the analysis, but that's out of the scope for this presentation. So lastly, we have a pharmaceutical group of users. So these are, of course, mainly industrial users, so pharmaceutical companies developing new drugs. But also in academia, there are enough groups that work with new drugs to identify which kinases are inhibited by the drug. And this can be either on targets or off targets. So this is part of target discovery, as well as pharmacological studies. So how does the drug affect the system? And is it better or worse than any other drug that you're interested in? And here we also would like to add that our PAMGene technology is a nice addition next to RNA-seq, for instance. So we've done some studies in the past that we do data integration of RNA-seq data or other omics data such as mass spec to get a bigger picture. So next to your more transcriptomic or proteomic data, we also add this kinase activity data, which can help develop novel hypotheses and conclusions. But to really achieve this, we have several different strategies here listed on the slide. So from our side, we generally try to organize symposia or seminars just like this one, where we talk about our technology and their applications. And what could also be interesting, especially for you, is to share local results. So if you have been running PAMChip assays in your institute, please share that you use the PAMChip assay, and then also, what did you get out of it? Perhaps it will inspire some people to run new assays on the PAM station. Along the same lines, you can also reach out to specific groups. So groups that would be interested in these assays. You can always approach them directly. This is also what we do. So we try, if we're setting up a new trial for a PAM station, we also attempt we also look at groups that might be interested in using the platform. There's also an idea to do workshops. So you can imagine if you organize a workshop inside the institute where people come to present your data, or you give maybe a hands-on demonstration of the PAM station, just to make people a bit more familiar with the technology, they will also start using it more. Sharing publications is, of course, a very important one. So we also have a dedicated section on our website where we have all publications belonging to all research groups that use PAMChip data. So you can really have a feel for how robust and how trustworthy our platform is. Plus, it gives you an easy overview of what's possible with our platform. So I mentioned seminars before, but next to this, you can also do marketing. So for instance, if you're really interested in improving the number of users in your institute, put up flyers, for instance, or talk to people about maybe having a short introduction presentation before a different meeting, just to ask people if they're interested in doing these type of assays. One-on-one meetings it's a similar approach as to reaching out to specific groups. So perhaps there's somebody at your institute who might be interested but doesn't really know where to begin. And then you can always go out there and say, hey, I've been using the platform for a while now. These are my experiences. Let's sit together and talk how we can help each other. And finally, doing pilot studies, or really small studies, just to give a feel of what's possible with the platform, is also a good way of attracting new users. So for us, the combination of these is really beneficial in getting new people to use the platform, also getting people to trust the platform, and hopefully performing more experiments down the line. So what we found also important is to compare our technology versus some different other technologies. So in this case, we compared it to reverse protein arrays, so the RPPA, Western blotting, which is a staple of any molecular biology or any biology lab really, antibody arrays, phosphoflow, so flow cytometric analysis of phosphoproteins, and as well as mass spec, or in this case, phosphoproteomics. And to really make this an interesting comparison, we looked at 5 different aspects of each technology. So at the top, we compare the sample input. So how much sample do you really need to add? In this case, the lower sample input you need, the better. The price per sample, of course, is very important for most people. How fast the run is. So in this case, a faster run is, of course, better. How high the throughput is. So how many samples can you measure at the same time? Is it possible to multiplex samples? And finally, the coverage. And in this case, this is the coverage of the kinome. And at the first glance, as you can see, our platform is quite good at most of these criteria. So out of all the technologies that we've looked at, we have the lowest sample input. So we need about 0.5 micrograms for PTK assay and about 5 micrograms of cell lysate for an STK assay. Compared to Western blotting, for instance, where you need about 30 to 40 micrograms per lane. So per sample, that's already quite a big difference. The price is generally is medium. So Western blotting in this case has the lowest price per sample, but mainly because you need to buy 1 antibody only, and the running of the gel is not expensive. RPPA is quite dependent on a large number of different antibodies, so this increases the cost quite rapidly. For mass spec, is also it can be quite costly to run 1 sample. However, mass spec does have the highest coverage of all technologies that we compare here, because of course mass spec can essentially measure each phosphocyte that is available in the phosphoproteome, while we can only cover about 377 kinases out of the 530, I believe, off the top of my head. However, this number is constantly improving. So with every iteration of our bioinformatics toolbox, this coverage number increases. Now, one important aspect that we don't show here is that, of course, our platform measures kinase activity, which is fundamentally different from phosphostatus. So I'll come back to this in a couple of slides as well. But of course, kinase activity really tells you when the kinase is active. And phosphostatus only tells you that the kinase has been active. But unfortunately, you can only do that for substrate proteins, so you don't know which kinase that is. So in our case, we can really show which kinase has been active. So this is the end of the finding new user sections. Are there any questions so far?
Nope. That's clear, Peter. Nice.
All right. Okay, now we can move on to some more assay-specific information. So these will be more points about how to prepare your best samples, how to run the best samples, as well as some tips and tricks for that. So let's start with some guidelines for the preparation of samples and the preparation, preparation of samples. So our preferred way of sampling preparation is how we do it in-house as well for our industrial clients, is to snap freeze your cell pellet or tissue and then perform the lysis. So really keep the samples as fresh as possible and as cold as possible. And what we generally suggest is to make several aliquots immediately before storing at -80 degrees. So the reason I'm saying this is that you might want to run the same samples for PTK and STK, but you'll also need a sample for protein determination. So that's already 3 aliquots that you need. And if you make them from the same tissue sample or same cell pellet sample, you're sure that the concentration of protein in each of these aliquots will be exactly the same. So as well, mistakes happen all the time. So I've been there before that my samples dropped out of my fingers and I couldn't use them anymore. If you don't have a couple of backup aliquots, you'll be very happy down the line. We can use paraffin embedded material as well, but be sure to use a maximum of 25% paraffin. A higher paraffin content will interfere with the assay, unfortunately. And do note that for certain cell types, you also require special protocols. So a very good case for this is the PBMC fraction from whole blood, so peripheral blood mononuclear cells. Because the way that we perform lysis is a little bit different than we do for cell pellets. We have a special protocol in this case. That's available from the resource, the PS12 resource page, if you haven't seen that. This is also true for perhaps for cell types that you haven't really used before. So most adherent and suspension cell lines will be fine. But you can imagine if you have some sort of rare cell population, like perhaps astroglia from the brain, or some very rare immune cell type, you might have to take special consideration into preparing your samples. We might have a protocol available, so it's always worth to send us an email and to talk about what will be the best strategy. But in some cases, it will also be necessary to perform some optimization yourself. But more on that in a couple of slides. And lastly, we would also let you consider to perform the lysis and the protein determination, so both of them, for the customer itself. So this generally reduces variations. It's also our strategy for samples that we process in-house. The big benefit of this is that lysis and the protein determination will always be done in the same way. So for instance, if you let your customers do the protein determination themselves, there might be an issue if they do the protein determination slightly differently than other people, and that gives you quite big variation. So of course, our assay is quite sensitive to differences in protein content. So therefore, we always like to stress that try to perform these steps in a standardized way, so that at least that part of the variation will be excluded. So to get efficient experimental design, there's always a consideration of which time point do you take for the assay. And an exact time point is highly dependent on your scientific question. So what we generally see is that for between 5 and 60 minutes, this will be like the ideal kinase activity range. So this is when your kinases will be most active after, for instance, an activation or an inhibition with a certain drug. And this is when you'll see the highest activity of your upstream kinases. So if you assume that you have an inhibitor against, let's say, EGFR, then it's probably worthwhile to do the assay after about 10 minutes after adding the inhibitor. So this is an important point that is sometimes a little bit confusing. But you can imagine that changes in signaling have an effect on the phenotype downstream. So changes in signaling will be prior to your phenotypic changes. And therefore, in some cases, it's worthwhile to look at kinase activity before you see any phenotypic changes. Of course, this is not really possible in all cases, and you have to do quite a lot of optimization to make sure that this really works. So what we also see is that many people run their samples about 1 to 24 hours after treatment. This is also because this is quite typical for Western blotting, that you do phosphoblot about maybe 6 to 8 hours after your treatment. And this is of course fine, but then the consideration is that you have to the results that you get might be many downstream effects. So these are kinases that are involved maybe a lot later than your direct kinase of interest. However, this gives you a bit of an indication of what pathway is active. Now, lastly, we also have a group which is higher than 24 hours, and this is typical for knockdown or knockout experiments, especially as RNA knockdown. takes about 24 to 48 hours before you achieve maximum knockdown. The same is also true for overexpression systems, in the case of transient overexpression. And basically the same sort of caveats are part of this time point as they are for the 1 to 24 hour time point. So there might be many you might see many downstream effects in your results. However, this doesn't mean that your essay is, of course, worthless. There's a lot of information that you can still get from the essay. My point that I'm trying to make here is that you should consider when your kinase will be most active. So another thing that occurs quite frequently is that if your kinase is not active in the first place, you will not see a difference in the activity of the kinase, because of course the PAM-CHIP assay is a differential assay. So next to considering when your kinases will be most active, you should also consider if your kinase will be the most active at that point. And another type of experiment that we do quite frequently, but we like to stress here in this presentation as well, is to use inhibitors directly in the assay. So as you might know, you can also add inhibitors directly to the lysate before you measure it on the PAM-CHIP. This has certain advantages. So you can measure much clearer primary effects of the inhibitor. So you can really see what is targeted by the inhibitor. You can look at really low time points as well. And you can also look at the contribution of certain off-targets, for instance. And another interesting aspect is that you can use not yet approved compounds on patient lysates, for instance. So you can imagine that if you have an unapproved compound that is a kinase inhibitor for the treatment of cancer, and you have a whole load of tumor samples from patients, that you then can immediately test them in vitro to see whether there's some sort of efficacy for your, at least on the kinome profile. But one important step here is that you have to optimize the spiking conditions. What I mean by that is that you have to look at the exact concentration that you add. So we generally suggest to do like a dose-dependent curve on the concentration of your inhibitor, so that you can at least have some inhibition of your kinases. This is partially due to having the kinases in a bit more dilute solution than they are in the cell. So the concentration of your inhibitor will not be directly comparable to the concentration inside the cell. But that's why we also suggest, okay, so run at least one optimization experiment with just a small number of samples, maybe N=1, just to get a feel for which concentration you need to use. So if you move on to the experimental design, this is also quite a frequent question that we get. So how do you really design the best experiment? How do you set up your samples? And basically there are in this case, I'm showing you two examples of quite typical questions. So for instance, you have one model and you want to look at 3 different treatments in the same, or multiple treatment conditions in the same model. So for instance, a low, a medium, and a high concentration of a certain drug, or different time points. Then the table on the left shows you an ideal balanced design for this model. So each column represents one PAM-CHIP, and each row is of course one array. So you have 4 arrays per PAM-CHIP. And on the first PAM-CHIP, this is the typical layout. So the first array will have a control sample followed by the 3 different treatments. And what some customers always prefer is that you randomize the position of your sample for the next chip. So the second chip, again, is a complete replicate of all samples, but then in this case randomized. And the same is true for the third PAM-CHIP. You can also, you don't need to randomize if you don't feel the need for that. We just prefer to give this as an option. But in this case, you have 3 replicates of the same samples, nicely balanced spread over the chips, which means we can also perform combat correction during the analysis and really get the best statistical result from that. Another option would be to compare differences between treatments in different genotypes or phenotypes, or different time courses with time mesh controls. So you can imagine that if you have, perhaps you want to see if the same treatment has a different effect in two different cell types. For instance, your first cell line is a cell line that has a certain kinase, and the second cell line is a knockout of that same kinase. Then this is the way that you would design experiments to compare the effects of that inhibitor on these cell lines. So if we again look at the first PAM chip, we have the control, so maybe the vehicle control for one cell line versus the treated cell line. And then we have the vehicle control for the second cell line and the treatment of the second cell line. And then again, for the second and third PAM chip, we have exactly the same samples, but then again in randomized. And this gives you quite a robust measurement. So this is the end of this assay design section. Again, I would like to ask if there are any questions so far before we move on to the next section.
No, Peter, no questions.
All right.
Great stuff.
So very clear so far. That's great.
Yeah, nice.
So let's move on to the typical results section. So, okay, you have designed your experiment. You go to the PAM station, you run your samples. What exactly can you then expect? So this is like really raw data that you get from the PAM station. And this is one example that we get. So let's focus on the left part of the slide first. So here in the bottom left, you see a raw image of the PAM chip. And you can see there's all sorts of spots that go up and down, so indicating the phosphorylation status of the peptides on the chip. And this can then be translated to one column in this heat map. So this heat map shows you many different samples. You see a lot of, for instance, high signals, but also a little bit of low signals. But overall, it's quite nicely distributed among all the samples. And what I like to stress here in this picture is that it is in focus. All the spots are clearly visible. There's a good signal to noise ratio. And most importantly, there is no obvious dirt or dust or any leakage or cracks. This is what a good PAM chip result looks like. So on the flip side, we have bad data. And this is an example of a chip that isn't in focus. So this can happen due to maybe slippage of the measurements, but it can also happen due to a leaking chip. And sometimes it can also be dirt on the chip, which is obscuring some of these spots. Overall, all sorts of things can go wrong. Now, what then happens is that you might not get enough signal, and you get this very obvious distortion in your heat map. So this sample, this image that I show you here belongs to this sample, which is completely different compared to the other columns in this heat map. And so this was enough for us to say, okay, well, this sample is an outlier, and this doesn't give us any data that we can use. So based on that information, we discarded this sample from the analysis. This is an important step to really look at quite strictly. So if you see a lot of bad data, for instance, you might also need to consider whether your PAM station is still performing correctly, or whether the experiment has been performed correctly. And we have a number of strategies that could help you to find this out. And I will give you a couple of examples in a little bit. But this is, because of course this peptide data is the beginning of the analysis. It has to be right at the beginning. And to facilitate this, we also have a peptide QC method. So if you've ever seen a report from us, this is also well described. And what I mean by this is that we have a sort of flag system indicated by the 3 colors here that also indicate the strength of your data. So if you have high quality data, the signal range will be quite large, from 0 to 4,000 arbitrary units, and a number of peptides passing quality control will be larger than 123 for PTK and larger than 90 for STK. Next to that, the coefficient of variation for the technical replicates will be less than 20%. For biological replicates will be less than 30%. And these values can go down. So if you have more variation and less peptides from passing the QC, as well as less signal, we can also categorize it as middle. So this will be like sufficient data quality or low data quality if it's even lower. So this also means that you have to consider your conclusions that you draw from this data. So high quality data will generally be fine for any data interpretation in later steps, but especially for low quality data or sufficient quality data, I would personally suggest make a small disclaimer saying, hey, your data is not as good as it could have been, but these are the conclusions that we draw. But be sure that this is not really, this might not be 100% true. So you should take care in validating this data and make sure that you really validate it properly. So this is generally how we perform this quality control step. We deliver quite an extensive report to customers where we describe also where something could go wrong. So an easy example, for instance, could be if you have a low signal quality, is that something went wrong during the lysis. So for instance, if you don't perform lysis on the plate, but you first release them with trypsin, release your cells with trypsin, then your signal intensity will degrade. Another option is that if you perform multiple freeze-thaw cycles of samples, your signal intensity will also degrade as the kinase will be less active in your sample. So these are all things to really consider. Finally, the last example I would like to share is that you will have cell types that will be more or less active, and this can also have an influence on your sample, as well as the protein amount that you load onto the chip. So these things are quite sensitive, and you can often benefit from optimization. So what you can consider optimization in multiple conditions, and in generally like normal your household cell lines. So think of HeLa or HEK or standardized model systems, so mouse or rat samples, for instance. These don't really need optimization, but if you're investigating a new model or new samples that you've never run before, it could be useful to plan an optimization experiment. You can also discuss with us how to approach such samples, because maybe we have run certain samples that you haven't before, as well as the example that I gave you a few slides ago about the PBMCs. And especially if you're running large experiments or experiments with multiple runs or like maybe 100 samples, it'll benefit to do a small optimization first. Now, to help you with these optimization experiments, we will also provide you with an exercise chip. So if you place a new order for new chips, we will give you a free exercise chip that you can use for such optimization experiments. So it's just a chip that has the same peptides spotted in different concentrations, and then you can measure if everything went well. And another application for this exercise chip is to really check the performance of your PAM station. So you can imagine that if you haven't used your PAM station for a while, you might want to check if everything is still in working order, if you still can get trustworthy data from it. And if that's the case, then please use one of our exercise chips. So as I mentioned before, we will deliver them to you for free. And the only thing we ask back is that you send us the results of your exercise chip back to us, and we can use that to improve our protocols. And so one way that we can measure this is that if you use the standard lysates that we ship with the kits as well, You can use that to run these samples. And I would suggest to do 3 technical replicates and then use 4 different lysates. So you have 12 samples in total. And then the result should be about lower than 10% CV on your samples. If that's not the case, then there might be something going wrong in the running of your samples itself. But this way, at least you can have a little look at if your system is still performing the way it should. You can also expand this, saying that, okay, I want to also improve my lysis technique. And in that case, you can also use the same cell line, but then lyse it 4 times. And each lysate run in 3 technical replicas and do the same sort of experiments. And that way you can really test every aspect of the instrument and your sample preparation. So again, this is the end of the typical results section. Before we continue with the frequently asked questions, are there any questions so far?
Let's look at the chat. Oh, no questions, so that's clear.
All right, then let's move on. So finally, I have a couple of frequently asked questions, and I think most of them I've already covered before, but this isn't just a short overview of some of these questions. And the first one we always get is how many kinases are covered? Well, at this point, we can cover about 377 unique kinases from the human kinome that can phosphorylate the targets represented on our PTK and STK chips. As I mentioned before, this number is always increasing as our bioinformatic pipeline is also becoming better. So this is by no means a final number. And what we often also hear is if you can use mouse samples on with our platform, because of course the peptides on our chip represent human phosphosites. However, the good thing is that there's about an 80% overlap between the human and mouse kinome. And approximately 30% of our users use mouse samples. Now, we haven't never really heard complaints from them, so this is going pretty well. But you should keep in mind that there might not the results that you get might not be one-to-one transferable between human and mouse. An important question that we often get as well is how many replicas to use. So in the examples that I gave before, I showed you 3 replicates. And this is the bare minimum for like a more statistical analysis as well. Especially for cell lines, this is an adequate number. But if you're using mouse models, for instance, or animal models, they are not as homogeneous as cell lines. So we routinely suggest 6 replicas for mouse models. If you're using human samples, so really from clinical samples, for instance, because the variation is much larger, we would suggest a minimum of 10 samples per group, just to make sure that you can have the most appropriate statistical analysis. There are some cases where you can use less replicas, but we don't really suggest it. More replicas is always better, of course, but you will need to run more samples in that case. We are frequently asked if you need positive or negative controls in the assay. And so because of the way that we do our analysis, we really compare between a control and a treatment assay. So in this case, the controls are already included. And frequently asked as well how we predict which kinase is active. And so I touched upon this before. So we use self-developed algorithms to predict active kinases from the peptide level data. We use databases that have recorded information of kinase to substrate relationships. So these include Phosphocyte Plus or UniProt. We have a lot more information on how we perform these analyses, which is kind of a little bit out of the scope for this presentation, but we can always refer you to more information there. And an interesting question as well is what kind of changes can we detect using the assay? And by changes, I mean like changes in activity between treatments. So in this case, we can detect quite small activity changes that range from about 20% or more. And then if you increase the sample size, you can detect more subtle changes as the overall variance sort of decreases. Now, finally, the last question, frequently asked question, is that if you can treat your lysate with kinase inhibitors. Of course, I went into this kind of quite extensively before, but you can directly inhibit the kinases in your lysate if you spike in an inhibitor and then measure the effects of this spiking differentially. All right, and then just to give you, before wrapping up, I'll just give you a little preview of what will happen in the next session. So the next session will be on the reporting of results. And then we would like to consider questions like, what do we deliver in a report for customers? What is our approach to reporting? And a difficult part is as well how far can you really go with interpretation? So we get these kinase activity profiles, and how far can we go interpreting these kinase activity profiles? And finally, we would like to teach you how to develop a process using a reports template. So in preparation for this, we would like you I would like to ask you if you could share by email your top 5 reporting related requests from your collaborators. So this could also be people that use your system internally. And then what kind of data would they like to see in the report, or what are they missing? So the expected date for this seminar will be January 2022, a couple of months from now. And of course, nearer to that date, you will get an invitation again. So that's the end of this initial session. Thank you for your attention. I hope it was at least useful for you, some of you. And if there are any questions still at this point, I will be happy to answer them.